Hi, I'm Amr Alexandari. I'm a PhD student in the Computer Science Department at Stanford. Today I'm presenting our work that demonstrates how neural networks, trained to predict transcription factor binding profiles and chip counts, can learn the thermodynamics of DNA-protein interactions. Transcription factors play a crucial role in gene regulation and underlie many different aspects of human physiology, immune responses, and cell differentiation. TFs can have a 1,000-fold or greater preference for specific binding sequences or motifs relative to other sequences. For many biologists, this high specificity implies an ability to regulate the genome. Yet there is only partial overlap between experimentally determined sites and motifs, and there is still a need for computational approaches to infer the sequence elements and associated TF binding events responsible for the chromatin accessibility at each regulatory element across the genome. ChIP-seq and ChIP-exo experiments have been used to quantify where transcription factors are bound in vivo, with all the complexity that comes with being in a cell. For example, cooperation and competition between transcription factors, chromatin accessibility, etc. At the same time, much research has been done to study protein-DNA interactions in vitro. In that line of research, we think about biological molecules interactions. The basis for interaction between transcription factors and DNA is a combination of noncovalent bonds, electrostatic, van der Waals, and hydrogen, and hydrophobic interactions. But much of the behavior can be accounted for by two biochemical parameters: the strength of binding, or binding affinity, and the kinetics of reaction, the association or dissociation rates. Since measuring binding affinities is important for a quantitative understanding of cellular mechanisms, many techniques have been developed to obtain these parameters. PET-seq has been used to quantify binding energies for transcription factors bound to naked DNA. In this simple system, we can measure thermodynamic quantities with high resolution. One way researchers relate the thermodynamic affinities from in vitro experiments to transcription factor occupancy in vivo is via position weight matrices, which are linear models that consider each position independently. Unfortunately, when these models are deployed, the performance is poor. We observe strong peaks without a good motif, and areas with motif matches that have no peaks, which leads us to a central and important question. Does this poor predictive power reflect the fact that all of the extrinsic cellular factors, such as other protein concentrations or chromatin accessibility, are required to specify correct in vivo localization? Or does it simply reflect the fact that our current models are not developed enough to accurately predict binding? The project is an effort to address this question. We trained BPNet convolutional neural network models to map 546 base pair DNA sequences from the yeast genome to base resolution PBXO and ChIP-exo profiles of FO4 and CBF1. Despite the small size of the training sets, which was less than 4,000 peaks, the correlation of predicted and observed counts across peaks in held-out chromosomes was on par with replicate experiments. Next, we used DeepLift to interpret the models. DeepLift is an algorithm that explains the difference in the output relative to what the output is under some reference input. It decomposes the difference from reference of the output into contributions from the difference from reference of input features. Here we have the prediction and the DeepLift scores for a region from a test chromosome. The model is learning the motif as evident by the visible E-box in both the counts and profile prediction tasks. We used the trained models to predict binding strength for millions of synthetic sequences which contained a high affinity core E-box motif, CAC-GTG, with systematic sequence variations in the plus and minus 5 base pair flanking the motif. To generate a prediction, we dinucleotide shuffle a random sample of 100 different backgrounds from test chromosomes. Then, the model predicts before and after the flanks and core motif are inserted at the center of the backgrounds. The difference in predicted log counts between the background before insertion and the background with flanks and core motif after insertion is saved as delta log counts for the sample. Finally, the deltas in log counts are averaged across the 100 backgrounds to produce a prediction. We found that these predictions from the PBXO and ChIP-exo models had remarkable agreement with the corresponding relative binding affinities inferred from in vitro BitSeq experiments. This figure shows the correlation for PBXO, FO4, and this figure is ChIP-exo CBF1 under ethanol-limited conditions. Next, we used simple linear regression to transform the predicted log counts to relative affinities. Note the similar scales on both axes and the high Pearson correlation. We used the inferred nucleotide importance scores using deep lift across all bound sequences to identify predictive motif instances and cluster them into non-redundant motif representations using TF-MODISCO. The derived TF-MODISCO patterns showed distinct flank preferences that match the preferences by average delta-delta-G from PET-seq. In this figure, we have FOV4, and we can see the clear G and C preference for the flanks. In this figure, we have CBF1, and we can see the G and T preference for the left flank and the A and C preference for the right flank. Our models now allow us to carry out large-scale in silico experiments. Earlier, we saw the GC flank preferences in FOV4. In this simulation, we slide a 30 base pair sequence of GC repeats closer and closer to the core motif from the 5' end. We observe a generally favorable effect on binding. In conclusion, we showed that neural networks trained to accurately map genomic DNA sequences to base resolution binding profiles of FOV4 and CBF1 from PBEXO and ChIP-EXO experiments in yeast can learn binding site flanking sequence preferences that strongly correspond to in vitro binding energy measurements from microfluidic platforms. Our framework enables a unified analysis of in vitro and in vivo TF binding assays via comprehensive in silico interrogation of deep learning oracles, which can be done at extremely large scales. Thank you.